So good morning class. Our next topic will be the separations and isolations of a DNA fragments. Okay, so in our previous classes we have studied about the nucleases enzymes, that is restriction endonucleases and restriction exonucleases. Okay, so we know that DNA fragments will be formed by the restriction enzymes, but in this DNA fragments will be especially formed by the restriction endonucleases, and that can be separated by a technique called gel electrophoresis. But now यो separation र isolation DNA fragments लाई हामीले कसरी गर्न सकिन्छ भने first of all DNA has to be fragmented. फर्स्टमा हामीले DNA को fragments बनाउनु पर्यो by utilizing restriction endonucleases. जब DNA को fragments हुन्छ अनि त्यो fragments लाई चाहिँ हामीले के गर्नुपर्छ, separate गर्नुपर्छ. larger fragment smaller fragments लाई separate गर्यो. Okay, separate गरिसकेपछि चाहिँ we have to isolate it, the required जुन चाहिँ हामीले DNA fragment चाहिएको छ त्यसलाई हामीले isolate गर्नुपर्छ. So यो separation र isolations of a DNA will be conducted by a certain technique. यसको लागि चाहिँ हाम्रो एउटा certain special एकदम sophisticated technique हुनुपर्छ, that is known as gel electrophoresis. Okay, the name itself suggesting that gel electrophoresis, that means a certain electricity will be given to a certain types of a gel. So in such gel this DNA has to be moved, this DNA has to be separated. Okay, we will study in detail. So after fragmenting this DNA, now this DNA fragments are, we know that DNA is negatively charged. Okay, अब यो negatively charged DNA लाई चाहिँ we can separate it by moving them towards a node under the electric field through a medium matrix such as agarose, okay, which is a natural polymer of D-galactose and 3,6-N-hydrogalactose, and which is extracted from a seaweeds. अब हाम्रो जुन चाहिँ त्यो फर्स्टमा कट गरेको DNA को fragments थियो, that fragments has to be like it has to be placed in a certain plate of medium called agarose gel. So agarose gel मा चाहिँ हामीले के गर्नु पर्यो, यसको matrix मा चाहिँ we have to place the these fragments of DNA. After that we have to provide electric field. Okay, त्यसको बादमा चाहिँ हामीले के गर्छौं, we are like this placing this DNA on the agarose gel matrix ma medium ma. After that we are applying certain electric okay, that is the electric field is applied गर्दैछ. Now this DNA fragment is a negatively charged, so this negatively charged DNA will be tends to like move towards the anode, that is the positively charged. Okay, and this medium or matrix is made up of agarose, and this agarose is the natural polymer which will be extracted from the weight. Agarose मा चाहिँ के हुन्छ भने that is the D-galactose and 3,6-N-hydrogalactose हुन्छ. So after that DNA fragments separate or resolves according to the size through sieving effect provided by the agarose cell. So यहाँनिर हाम्रो medium मा चाहिँ there will be the agarose gel. यो agarose gel को कारणले गर्दाखेरि चाहिँ this will have a certain sieving effect will provide. So after that this DNA will be like separated according to the size. That means the smaller size of fragments move faster. That means जो चाहिँ small को size छ एकदमै fast move गर्छ in the matrix. Okay, and the separated DNA fragments can be visualized after staining the DNA with ethidium bromide. र जुन चाहिँ agarose gel मा भएको DNA, this is not so visible हुँदैन यसलाई चाहिँ हामीले भिजिबल बनाउनको लागि चाहिँ वी हेभ टु प्रोभाइड सर्टेन स्टेन अथवा डाई द्याट इज द कलर प्रोभाइड गर्नुपर्छ द्याट स्टेनिङ अफ द डिएनए विल बी गिभन बाई द इथिडियम ब्रोमाइड दिस इज द स्टेन एन्ड फलोड बाई द एक्सपोजर टु द ultraviolet radiations so bright orange colored of a DNA bands can be visualized okay after this pouring these DNA fragments in the matrix in the medium of the agarose gel and after providing certain electric field these DNA fragments will be forced to separate it or forced to move towards the anode according to this their size that means the smaller size of the fragments will be move faster as compared to the larger fragments so after that this will be added with a certain stain or a certain like dye okay and after that this will be exposed to a certain ultraviolet radiations and after that ultraviolet radiations लगाइसकेपछि चाहिँ certain brown color structures brown color fragments will be seen in can be easily seen okay and after that these DNA bands so you can see that this is the bands of DNA okay these all are DNA so this will be seen only after the ultraviolet radiations को exposure को बादमा चाहिँ अब DNA को bands हरु चाहिँ separate भएको छ that will be cut out from the agarose gel एगरोस जेलको केमा छ मिडियममा छ यो जेलबाट चाहिँ कट गरेर बाहिर निकाल्छ will be extracted out from the gel piece and this step is known as elution so elution भनेको चाहिँ it is a it is a step in which this DNA will be separated and will be separated after cutting out from the agarose gel okay and that will be used for the further research okay these purified DNA fragments are used in constructing recombinant DNA by joining them with the cloning vectors so in our previous class we will be talking about कि recombinant DNA जुन चाहिँ DNA लाई चाहिँ हामीले केमा लगायो another type of like DNA मा join plasmid मा join insert गरेर चाहिँ that will be again inserted into the host organism after that in the host body this will be multiply in a numerous number मा multiply हुन्छ एउटा purified DNA भनेर हामीले स्टडी गरेको थियौं so purified DNA fragments will be formed after these performing gel electrophoresis techniques okay so you can also see this pictures has been taken from the textbook okay so you know that this one is the larger band of the DNA this is the smaller band of the DNA okay so this shows that a smaller band of a DNA is in the bottom because this will move faster because of its smaller size as compared to the larger bands of DNA okay you can also see the same thing in these pictures also so there will be the setup in the gel gel electrophoresis that is the agarose gel electrophoresis there will be a certain lane okay lane 1 2 3 lane will be there okay so where these mixtures of DNA molecule will be inserted okay there will be the medium gel cha यसलाई चाहिँ gel glasses plate cha so यो gel भित्र चाहिँ के हुन्छ agarose gel हुन्छ this is the natural polymer यहाँनिर चाहिँ के भयो अब mixtures of DNA molecule हामीले insert गर्यो त्यसको बादमा चाहिँ we will provide the electric field provide गरिसकेपछि now this DNA will be start moving after providing electric field because DNA is the negatively charged molecule so this will tend to move towards the anode or towards the positively charged molecule so the smaller जुन चाहिँ सानो particles हुन्छ that will be penetrate that will be move faster according to the sieving effect of the agarose gel ma okay so after that certain bands of a DNA बनिन्छ this is the bands of DNA that can be seen in the gel electrophoresis. So that means gel electrophoresis भनेको चाहिँ it is a technique in which the gene will be separated according to its regarding to its size, and we can be able to get a purified form of DNA, and that purified एकदमै pure DNA fragment चाहिँ हामीले further constructions of the recombinant DNA को टाइममा we can utilize, so that त्यो small parts of DNA लाई चाहिँ we will be joining with the DNA of the certain like vectors, अनि त्यसको बादमा चाहिँ multiplications of our desired DNA will be occurred.